The overall goal of these procedures is to adapt specific methodologies for the determination of mechanical properties such as stiffness, viscosity, and adhesion to the specific case of embryos and developing organisms.
The methods that we introduce in this paper can answer questions in the mechanobiology field, such as how material properties may respond to genetic or physical inputs during development or impinge in the regulation of cellular activities. The main advantage of these techniques is that they allow retrieval of crucial information with minimal manipulation, addressing questions that are unavoidable by other procedures.
These methods provide insight into understanding the role of the yolk cell during epiboly, and then they can be applied to different domains in the embryo, to different developmental stages, and even to other model organisms.
Visual demonstration of these methods is critical as embryo mounting and handling are difficult to learn.
To begin, collect zebrafish embryos and grow them at 28 degrees Celsius in E3 embryo medium. Stage the embryos according to morphology. Manually decorionate stage zebrafish embryos of different ages according to individual interests under a dissecting stereo microscope with an adjustable range of magnification between 8 times and 50 times. Remove the chorions with 2 thin and sharp forceps. Grip the chorion using one set of forceps and make a tear in it with the other set of forceps. Then, holding the chorion in a region opposite of that of the tear, push the embryo gently through the opening. To mount the embryos for AFM, first fill a 30